All right, so we have our groups, as you know, and then we were kind of just going to the table and ended up writing some stuff on there. So I may look at the pictures on the groups, and I mean, if we have time at the end, we can come back and write some notes in there. But as we mentioned, the notes in the slides are pretty good outliney wise. The main things you need to know for the groups, though, like I'm not going to go into massive detail about all the ins and outs, but it would be things like, actually, I'll show you this. I put on here, if you follow me, you'll see the learning objectives. Actually, let me just go up here first. I copied and pasted them from the learning module, which I also embedded in here. So it's on D2L, but I also embedded it in here. The learning module is in the AMP section, I think. But I can put a second copy up here too. But you can see the animal basic characteristics. So we've talked about all that. The origins we've talked about. And then the diversity of animals or the different groups, which is actually the boards down below, but I also put it here. So you can see the kind of stuff you need to know for the different groups are whether they have true tissues or not, the symmetry, sort of what kind of coelomate they are, advantages of the coelom, which we already talked about, circulatory system, just whether it's closed or open or they diffuse across the body wall, for their gas exchange, functions of the circulatory system, which we'll touch on, respiratory, etc. So you can see the different kinds of systems. And after these, things in parentheses, that's like the basic thing that you want to know about them. I'm not going to go into massive detail about describe protonephridia, but you would want to know that protonephridia are in the flatworms, which are the Platyhelminthes phylum, as we shall talk about. Okay, so just thoughts about that. And then we can come down here. Okay, so this is on the animal phylogeny board. So what I did do is to put the learning module in there. It has lecture videos embedded. I also specifically put the animal phylogeny lecture separately so that so it's right in there, to watch, because of course you'll be responsible for the phylogeny, which is less than the phylogeny that we have with the papers. So the papers up here have more groups than you're going to be responsible for. And the groups you're responsible for is the groups we're talking about in the animals tables that we're filling out and the ones on the board. And they're the ones that are in your book. Or I'm going to say book. Some of them are in the book, but then they're all in that supplemental packet, the supplemental chapter 33 reading thing. Okay, so this is the same learning objectives as above, but I just copied them down here because that's where they're relevant. Okay. All right, so we'll just pop over to the animals tables again. I think this is an efficient way of getting through the groups. And like I said, I might go up and look at the pictures of the different groups to make sure to give you an idea of what they are looking like. Okay. So I also realized we needed to add just a couple of, another thing to the ctenophores and cnidarians with their feeding structure kind of characteristics. These ones in green are more than one group has these characteristics potentially. And so yeah, you can see there's doubles of some of them. So most of these groups are going to have, Porifera has no true tissues. So we talked about the digestion there. Then we have a few groups here with gastrovascular cavities, which are where you have just a single opening into this pouch. The pouch does the digestion, and then the food, or after it's been absorbed, that whatever's not absorbed or broken down and absorbed comes back out of the same opening, the gastrovascular opening there. And so we'll need to put this in Ctenophora. They do have a little anal pore, but it's, I don't know, they don't consider it a complete digestive system. So mostly the food's going to come in one entrance and go out the same entrance. So that's a gastrovascular cavity. Not a lot of specialization along the way there because it's just everything's happening in the same spot. Same with cnidarians. They have a gastrovascular cavity as well. If we go over to the, let me find it, phylogeny here. Look at this one. What we're going to notice now is that we're in bilateria, and all these groups are going to have that bilateral symmetry. They're going to have the triploblastic development, where they have the mesoderm layer turning into the muscle. They're going to have cephalization, where they have the some nervous tissue more organized at the anterior with sensory structures at the anterior, which allow them again to do the hunting, avoiding being prey, directional movement, that kind of thing. With the bilateral symmetry, of course, we have like, you know, head, tail, anterior, posterior, left, right, and then dorsal or back and belly or ventral side, which we talked about at the beginning of the session of the unit. So you'll notice here they're split into kind of two groups: protostomia, which some people use, some people don't. Not all protostomia does protostome type development. Deuterostomia is definitely a group. People use that name, and they all do deuterostome type development. But there's some in the protostomia that kind of have deuterostomy type development, at least partially, like the arthropods and some annelids and such. But for sure the book does use deuterostomy, ecdysozoa, and lophotrochozoa. And you'll see, where are we here? Here, we're starting at Platyhelminthes. We're starting in this lophotrochozoic dysozoa, so actually lophotrochozoa group here. And find my pens. All right, I think it just slipped underneath something. Oh no, I put it in the holder. Okay. Okay, so we're starting with, this is a group to know, Platyhelminthes. Actually, let's do this in a different color. Let's do it in lophotrochozoa color. We'll make that this pink color here. Okay. Okay. And so you'll notice that even though I'm doing it in this order, it's not like they're the first group to diverge after Cnidaria. So Cnidaria are down here. They are the sister group, as far as we can tell, to Bilateria. But they're not bilateriates. The rest of them are. And you can see, like I said last class, the dotted lines means we're not sure. And then there's a lot of polytomies here. You can see where they're all coming off together, because there's some uncertainty about that as well. So they're not obviously like all diverging at exactly the same time, but it's like, okay, we know these are related, but we don't know exactly who's most closely related. So let's just draw polytomy. And for our phylogeny, a lot of it is polytomies. And then let's see. So yeah, so Platyhelminthes, it's not like when you're drawing it on the tree, it's going to be like the next step diverging. It will just be within the Lophotrochozoa group. And actually, you know what? We can quick. I think we have time to quick draw this. Just here. I'm not going to go into massive detail about it, but this is what you'd want to. How we're organizing this deal. Okay, so we have, it doesn't matter what order you put these first three in, right? Because they're a polytomy here. But we'll just do porifera, and then we'll do, prettier, Ctenophora, the comb jellies, and then we'll do Placozoa, those little cup-like animals, the quite small ones, and then we'll do Cnidaria, the stinging jellies and corals and sea anemones and sea fans and such that have the tentacles that have those cnidocytes, which is stinging cells. Then we have here, this Bilateria is right here. So Bilateria is the, oh, this is an important thing. So Bilateria is the, that's not actually that. This is too early. Bilateria is the group. The synapomorphy is like bilateral symmetry, triploblastic development, etc. So if I ask for what group are they in, you're saying Bilateria. If I ask what's the synapomorphy, what's the characteristic? Well, Bilateria is not a characteristic. Bilateral symmetry is the characteristic. So we'll make that the big characteristic here. Actually, you know what? Mess this up. I need two branches, sorry. Here. This one's going to have 6 groups, I believe. that we just do 6. 1, 2, 3, 4. All in a polytomy, 5, 6. This group, we're just going to look at 3 in a polytomy here. And then this group down here. Oh, we're going to look at just Hydrostonia. We are going to branch it there. Like this. Okay. Good enough. Okay, so now we're on the Platyhelminthes. If you hear the word platy, is there anything that comes to mind? You can turn on your mic and say something. I'll monitor the chat as well. Platy. I see somebody. Let's see. Platypus. Yeah, that's something that I think of too, right? Platypus. Platypus, I think most people are familiar, but it's that mammal that has the duck beak looking thing. It also reminds me of the word plate. That's the same thing. So the word platy, and we'll go back down to our little table here, actually means flat or broad, but we're going to use flat. Okay, so platy means flat, like the flat bill. That's why platypus is named after its flat bill. So Platyhelminthes their common name is a flatworm. Does this work? There we go. Okay, so platy meaning flat or flatworm. And another common name we could use here is the Platyhelminthe. Platyhelminthes is the phylum. Platyhelminthe would be a common name for it that's not capitalized, or flatworms. Okay. Oh, and Nidaria should be Nidarians. That's the common name. Different, Ctenophorus, Ctenophora. Same kind of thing here with Platyhelminthes. They are in metazoa, true animals, meaning they have true muscle and true nervous tissue. They're in metazoa, meaning all animals, DNA, and the collagen layer and types of junctions. Eumetazoa, that's the one that means true tissues. It's specifically for animals, muscle tissue and nervous tissue. Bilateria, yes, they are. This is the first group we're talking about in bilateria, so I'll mark that with our blue color. And that means that bilateral symmetry, triploblastic development, and cephalization. Okay. They are in, they are in the Lophotrochozoa spiralia group. So I'll mark that here. The group is mostly characterized, like the difference between Lophotrochozoa spiralia and Ecdysozoa are mostly DNA difference. It's pretty clear for a lot of the animals which group they fall into DNA wise. Some of them are sort of like out there, and that could be because they evolve very quickly because of selective pressure, or very unusually slowly, so it gets a little tricky to place them. You saw those with the dotted lines on the phylogeny. Lophotrochozoa is named after some features that it has during development in the lifetime. The Lophotrocho, or the Lophophore, which some of the organisms have this feeding structure, and the Trochozoa developmental stage. I'm not too worried about that. Spiralia is the name if you're referring to like the spiral cleavage, because these are all, almost all of them, or many of them are protostomy type development, which has that spiral cleavage as we talked about. But if you know they're in the Lophotrochozoa group or Spiralia group, that's good enough for me, mostly separated by DNA from the Ecdysozoans, which are the two groups of bilateriates we're talking about. There is a third group, and that's animals too. That's the Deuterostomia, which I don't have on here, because we will make a table for all the Deuterostomia similar to this later in Unit 5. All right, so what are the defining characteristics of the Platyhelminthes? Let's go look up at our cards. This one here, right? So worm body plan. DV means dorsal ventral flattened, no coelom. So dorsal ventral flattened means the back and the belly are real close together. So it's like flattened along from, the back and the belly. So this does make sense that it would be the flat worm would be Platyhelminthes. Yep, that would have that characteristic. So that's the classic. Now, what's a worm body? A worm body means that you have like a long kind of tapered body with a mouth at one end and the anus at the other end, just like on the ends of those structures, like our earthworm. Earthworm is not a platyhelminth. You noticed it was kind of, it was not flat. So that is indeed what we're looking at here. There's a number of worms. They're not all in the same phylum. They're not all closely related. But all the flatworms are. So dorsal ventrally flattened, no coelom. So remember that coelom is the body space inside the body cavity. These guys are what we call acoelomate. So we could write that in here as well. So no coelom means they're acoelomate because A is like without. Like asexual reproduction is reproduction without sexual reproduction. Atonal is music that doesn't have like a key to it. So acoelomate means they don't have coelom. So if we come over to the little picture, pop over here, to our platyhelminth group here. If you look at this body plan here, the body layout, you'll notice that they took a cut through and it's all a big block of tissue, right? You see a space in the gut tube, but you don't see any space in the body cavity. That red mesoderm, which is now muscle, takes up the whole thing. So there's a few groups that are acoelomates. Platyhelminthes is one of the groups. Now they used to categorize and classify, make their tree of the animals based off of what kind of coelom situation they had. So they put all the acoelomates together, all the pseudocoelomates together, and all the eucoelomates together. But then once we got the ability to do DNA stuff, we're like, oh yeah, that's not correct, or highly unlikely because they just don't group together like that. But so now we just use those to describe the body plan, just like prokaryote, eukaryote. Prokaryotes aren't actually more closely related, the archaea and the bacteria, but we use it still to describe the cells. Here, acoelomate just means without a space. So these guys are not going to have the advantages of a coelom, but it works fine for them. They're generally relatively small, like planaria are like really small. Other flatworms, so here's an example of a flatworm that's on it's either a coral or a bryozoan. But it's bigger than like microscopic, but they're not super big. Flukes are real small. Tapeworms can get very long, but their body is pretty small, and each segment is small. So they do a lot of diffusion across their body wall, which we can write in a minute. They're the ones that I was talking about, planarians at least, not all flatworms necessarily, but planarians, the ones that regenerate their body. So I put a real short time lapse in here of the body regeneration. If you want to look at that, I know I don't think I can play it for everybody, but you'll see it has, it grows the head back. So they cut it in parts and then it grows the head, it grows the tail, it grows both sides. And so it can regenerate there. But you'll notice that flattened body, and we'll come back to that in a moment. So let's talk about the types of subgroups we have here. Oh, I should say, do I have a picture of this? Sorry, I'm zooming a little bit, but I don't have this up. Let's see if there we go. So I'm just sticking on the Platyhelminthes board a little picture of its gastrovascular situation. Okay, so I've just popped a little new picture in there. So you'll notice here this brown parts, you can see it's labeled gastrovascular cavity. So all this brown branching part here, that's the gastrovascular cavity. So it does have some structure to it to increase surface area, which makes sense because more surface area, more absorption across it. So the more food it can get into its cells. It can mostly do all this, like everything via diffusion and absorption and that kind of thing because it's so flat. So it doesn't have too many cell layers to try to get the stuff between. And so they're calling it a mouth here, but really it's the mouth and the anus because it's just one opening. You'll notice it's actually in the middle of the body. So, you know, just because ours is in our anterior doesn't mean that's how everything is, the animals. But I would call it a gastrovascular opening or GV opening truly because food comes in and out there. They can actually do reproduction through here as well. I think the male stabs the female and then deposits the sperm into the female body. It doesn't permanently hurt her, but it doesn't sound particularly fun. But that's how that works. Now this is for free-living flatworms. There are also groups in here that are parasites. So two of the groups of Platyhelminthes are parasites. They actually have kind of a totally so flukes have GV cavity. Tapeworms have lost a lot of their body parts actually because they live in the gut. They're able to get food straight across their body wall because their host already digested it. So they don't even have a gastrovascular cavity because of their parasitic nature. And you'll notice that often is that if something's a parasite, often it's lost a lot of body systems depending where it lives, particularly digestive stuff, because it's not advantageous anymore. It's just extra energy and body parts to make, and so it will be selected against. Usually what parasites have a lot of is reproductive tissue, lots of reproductive stuff going on because that's like mainly what is going on. They don't really have to support themselves except for making more of the worm in this case. So we'll come back down here. Let's talk about to our little table. Let's talk about some platyhelminth groups. Okay, so some subgroups. There's 3 subgroups. I'm not worrying about the official names. I know the book uses like rhabdo something, or I can't remember exactly the name, but I'm not worried about that. I'm just worried about these kind of common groups. So one group we have is would be the what's going on? I'm going to call them free-living flatworms. What free-living means is basically non-parasitic. And these can group with some of the parasitic ones, but they're non-parasitic flatworms, like the planarian. So these guys are just eating their own food. They're usually swimming, and I guess most of them are in water. There are some on land. Actually, I see them here. They're called hammerhead flatworms. I've seen them before. You might have seen them too. They are quite flat. They are out on land, and they have this like, looks sort of like not quite like a hammerhead shark. I'll put a picture up of them, but they're not good for the environment. They kill a lot of stuff. They look like this. Little worm body like that. Has anybody else seen those crawling around? I see them on like driveways and things. Or maybe even like this. They're longer body, something along those lines. I'll put one up though. You can keep your eyes out for it. You might have thought it was just like a weird earthworm or something, but yeah, in fact I believe it's called a hammerhead flatworm. But they're serious predators and can kind of damage the ecosystem. I believe they're an invasive species in the Houston area. But those are the free-living non-parasitic flatworms, like our planarians. Then we have the parasitic flatworms. I guess we can say free-living because we know it's flatworms. Free-living versus parasitic. The parasitic groups are the flukes and the tapeworms. So one of the essays that you're going to have to write about is economic impacts of a variety of groups. So if you use infections, you'll need examples of infections. They could be from any of the groups, but that's like one category. So you can give a variety of infections, but that still counts as like one thing that the animals impact. Of course, you can use, as we'll see later, there's foods. We eat some of these groups, so that would be a good one. We talked about sponges with their anti-cancer properties. There's actually some other groups that make molecules we might be able to use as well. There's silk stuff that we studied from silkworms and from spiders for tensile strength and building our own materials, and a number of other economic uses, but you'll want to gather a number of examples of those so that you can write about them. So what are the parasitic ones though? Flukes, they are carried mostly, they have these complicated life cycles where they have hosts. One host for some types of flukes are snails. And the reason I mention this is because there's lots of snails in gardens. So if you're someone that gardens, it's good not to touch the snails usually, because some of them, you don't know which ones have fluke inside their body. They don't hurt or fluke eggs and things or stages of fluke development. But the flukes can get in your skin, especially if you have a cut. But if they find an opening, the transfer could happen from the snail into you, and then you get a fluke infection. I actually had a student, she was a big gardener, and she ended up with a fluke infection once. You can get them otherwise. But yeah, if you don't play with the snails, only certain species have that. Then there's other more complicated ones. So fish, if you're a fisher person, freshwater fish can get fluke infection, so you have to be careful of that. And they can be carried by other things like, I know there's some species in ducks, there's some species in, those are the ones that, maybe frogs. Those are off the top of my head though, but snails are a really common one that we would encounter in our daily lives. That's one of them, flukes and fluke infections. And then tapeworms. You can get a tapeworm infection that's in the gut. Flukes can infect, there's like liver flukes, there's lung flukes, so they can get into a number of different tissues depending on what type they are, maybe even more than one in the body. But tapeworms are in the gut, in the intestine, and they're basically absorbing your food, so they can actually make you lose weight, but in a dangerous way. There was some wild time when people were using like tapeworm infections on purpose for weight loss, but that's like a really bad idea, obviously. But yeah, then they have they have these little body segments, so they almost look like segmented worms, but they're not segmented worms. They are, which we'll see, are like the earthworm, because they're very flat, and their segments are just these body, like each one is an entire body unit, if you will, and then they just break off the body units basically into these cyst forms. You poop them out, and then they can get taken in by other individuals and infect them. So that's, the tapeworms get passed like that. I can show you, I'm putting a picture of a tapeworm onto our slide. So if you're following me, you can see that here. Here's your little segments. Each of these is a little body segment, but not a segmented worm. It's a flatworm, and basically it's reproductive stuff. They even have some sort of suckers and hook to hook onto the intestine. So like because it'll come out the anus. So some people will be like, oh, if I just pull this out, I'll get the worm out. But it actually just breaks the body because it's anchored in there, and it would continue growing. So you need to get medicine for treatment for tapeworms. So osmoregulation is ways to balance water salt concentration, so your body's not exploding, because too much water and the body is not swelling or exploding cells, and the body is not like getting super dehydrated. So it's also important because you need the right combination of ions and such to be able to like do nervous system like signaling and other kinds of transport across the membrane of those ions, so that's important as well. For these Platyhelminthes, actually, should we go look Protonephridia? Let's do that. All right, so I'm going to come back up here to the flatworms. So Protonephridia are the osmoregulatory structures. Oh, that's this. So you can see there are a bunch of tubules, again, lots of surface area, which makes sense because the more surface area you have, the more exchange you can do. In this case, it's water and ion exchange. And they have these little flame bulb cells and all these tubules. The tubules have openings into the body wall, and these little flame bulb cells are going to basically they have cilia. They sweep in the water or the body fluid. That's what interstitial fluid is. So they sweep in the fluid in the body. There's going to be exchange across there. to retain enough water but not too much, and to keep the ions in you want and get rid of the ions that you don't. And then basically the waste is going to go out the body wall through that little opening there, and whatever you need still is going to be retained in the body cavity. So it's a bunch of diffusion here, but these are called protonephridia. That is the type of excretory structure, not excretory, osmoregulation mostly, that the Platyhelminthes have, at least the ones that are free living for sure. I think flukes as well. Like I said, the tapeworms are modified because of the parasitic nature of their life, but protonephridia are for sure. Like if I talk about that, it's a flatworm thing. Okay. And that helps with water regulation. Their body support system, a lot of these groups are just going to be a hydrostatic skeleton. It's not a hard skeleton. All it's doing is using water pressure in the body to keep it from collapsing. So that's like osmoregulation helps it keep its hydrostatic skeleton at the right plumpiness, if you will. And so that's what it's using for support in these. They don't have any hard structures there. No segmentation. We'll just leave that blank. The excretory product is mostly ammonia. So excretory, you can make a note of this as well. A lot of people think excretory excretion, like excrement, because sometimes we use that to mean poop or, feces, stool, whatever you want to call it, digestive waste, in other words. But excretory actually is referring to nitrogenous waste, which is like urine in humans, kidney stuff. So let's just make a note of that. The excretory system is getting rid of nitrogenous waste, which means nitrogen, like nitrogen-containing molecules, and that's from protein breakdown, primarily. So I don't know if you remember from Bio 1, but when you do cellular respiration, which is why, a lot of what the food is used for, it only uses carbons and oxygens and hydrogens in the cycle. There's no nitrogens involved. So when you actually take in proteins which have amino groups on them, which you might recall are these nitrogen groups on the amino acids. This is just like an alanine amino acid. You take off the nitrogen groups for processing, deamination it's called. But that's before it, like the rest of it will go into cellular respiration. But the groups that are nitrogen will be taken off. We'll just leave that. And if they build up, they would be toxic. So we need to get them out of the body. They're one of the body waste products. CO2 is another waste product from cellular respiration. The undigested food is a waste product, but these nitrogen groups are waste products. So the first thing that they come off as, straight up, is the NH3, which is ammonia. And that's pretty toxic. It's really easy to make because you can see it's just like one more hydrogen. That's like the process of breaking it off. So it's low energy to produce, but it's highly toxic. Generally, you need a lot of water to be able to dilute it to get rid of it, and so it doesn't have toxicity. So you'll see a lot of ammonia in organisms that live in water because they can dilute that, particularly fresh water, but also perhaps marine water. So that's a common one you'll see in a lot of groups because it's easy, especially if they live in water. Another product that ammonia can be turned into, ammonia can be turned into is urea. So it still has the nitrogen in it. It's just a little more, it's a little larger. have this in here? Maybe not. Okay. I can look up the structure of it and put it in here, but it has the N nitrogen group. It's just more energy to make. So you can convert ammonia to urea. More energy to make, but it's less toxic. This is what we have. Our ammonia is converted into urea. That's what comes out in urine as your nitrogenous waste, along with excess water for us and salts like ions. Less toxic, which means you lose less water. Okay. Okay. But you still do lose some water. So you can actually also convert ammonia to a third product, which is called uric acid. We'll actually see this a lot more in the animals too, but it applies to animals 1 as well. Uric acid is, it has like a ring structure in it, so it's pretty complicated to make. So it takes the most energy to make, but you lose very little water excreting it. It's actually like, it's a paste or solid. So this is like in birds, when they poop, it has that white stuff in it. That's uric acid. Or reptiles, if you've ever owned reptiles, sometimes they put out these little like dried white crust, like little ball things. And those are your uric acid. So you lose very little water. So we see these in a lot of organisms that are like living on land. Urea can be living on land too, but uric acid is particularly adaptive. So for example, we see these in land snails, and arthropods are going to be using uric acid. Particularly land arthropods. There'll be spiders, insects on land. Most animals in animals 1 are going to be getting rid of this as urea. I mean, not urea, ammonia, because they live in water. So let's add that. Many of the ones that are going to excrete it as ammonia are water-dwelling animals. Not all, many of them. But I don't think there's anything on land that's going to, or completely on land that does ammonia. It's too toxic. So this would be, there's an essay about insects moving to land and their adaptations of arthropods, particularly insects, to living on land. So using uric acid as the waste product for nitrogenous waste for the excretion of that would be a great example of one way that they have been able to move to land, insects, because their nitrogenous waste product does not lose a lot of water. It's pasty, very little water loss, which is great for conserving water for living on land, because living on land requires water conservation as one of the challenges, basically like something had to evolve in order for them not to lose so much water. Otherwise, they never would have moved onto land from their water-dwelling ancestors. So this is a good example of that for the insect essay. Because nearly all insects live on land. Very few other of the groups we're talking about live on land. Some like snails, some slugs, which are mollusks. Spiders, obviously, which are part of arthropods, but they're not insects. Millipedes, centipedes. So lots of arthropods, I suppose, live on land, but insects are particularly well adapted. So excretory product for Platyhelminthes. A lot of them have used ammonia. Some use urea. When I ask you these directly, I think the most important ones are the Arthropoda, really. And some of the mollusks. The idea here, though, is the land-dwelling versus the water-dwelling. And then we said their digestives is a GV cavity. They actually think, so originally they thought Platyhelminthes was like the first one to branch off because it was like, ooh, look, gastrovascular cavity, that's like cnidarians, so it must be early branching. And then the alimentary canal, the full digestive system arose. But now they don't think that that's true based off of DNA and a lot of other evidence. So it seems like probably this full digestive system, which we call an alimentary canal, was in the ancestor of all bilateriates and then probably lost in the Platyhelminthes group. That's what it seems to be, that it was a loss in there. Nervous system, they've got a nervous system, just ganglia, not a real brain in their head, of course. And these have a ventral nerve cord. We're going to see that that's most of the groups have ventral nerve cords with ganglia, unless they're cnidarians or ctenophores, which remember have those nerve nets. And I'll come back and fill this in. We might not have time to fill all these in, but I can come back and fill it in for you to have like a kind of collated, chart of all the kinds of characteristics that you kind of want to be familiar with or know directly for the anatomy physiology part of the exam. Okay, I think we should focus now on the characteristics, this chart, characteristics examples, and then maybe I'll do insect adaptations to talk about some more of those since that's one of your essays. All right, so now we have a group called Syndermata in your packet. I believe they still call them rotifera, but now they call it Syndermata because it's rotifers plus a few other groups, mostly rotifers in this group. That's the one we care about particularly. So they are really quite small. They're going to be microscopic. And, they don't really have another nickname besides rotifer, but that's not equivalent to Syndermata. But if you just know rotifer, that's fine. They are eumetazoans, actually, so we know all of these are eumetazoa from here. Okay. They are in bilateria. That's also true for the rest of these groups. Continue on that. And in this case, they're the Lophotrochozoans. Okay. So if we come here, they are very small. This is the only one that talks about small. They have ciliated feeding structure on their head. They call it the corona. Basically, it sweeps the food into their mouth, and then it's going to come out the other end. They have a full alimentary canal now, the digestive system with a mouth and an anus. I would mention, because you would want to know this, when you have a full digestive system from front to back, like our earthworm, like our crayfish, and I did mention this when we dissected, but it allows specialization along the way. So it's like you can have the pharynx, which takes the food in and like moves it down, does a little bit of softening there. But then you have like the crop or stomach, something like that, which is going to be softening. And then the gizzard, which is grinding, or that could be done in the stomach. So you have a bunch of different kinds of, and then the absorption happening in the intestine, the absorption of food, the absorption of water, and then the probably absorption of water at the end, and then the release of the waste products. So that's not just going to be digestive waste, but potentially also nitrogenous waste, depending on the organism. So, so this digestive system is specialization. That's the advantage of having a full digestive system, which rotifers do actually have, even though they're very, very small. Let's put this over here. And you can look at the picture of the rotifer on the slide. Maybe I'll pull it over here later. We don't need to know any groups of that specifically. So that's kind of what you need to know about rotifers. Brachiopoda. So these guys, I'll come over here and show you a picture. Here's the rotifer just for, it's little, very small. So this is under a microscope. So you can see those cilia, and then the water and food, particularly the food, is going to just get swept in and then go through digestion and stuff, and then go out the anus at the end. So this would be called a complete digestive system, or I've mentioned this word, alimentary canal. It's another name for a complete gut, mouth, and anus. Versus a gastrovascular cavity. The Brachiopoda or brachiopods are these, like, neither of these really have a stalk, but they're like, they look like a clam on a stick or a clam on a stalk. So these are the examples of Brachiopoda. So brachio is referring to arm, and we know poda is referring to foot or leg. So, because I don't know if you know, but like brachial artery and stuff is your arm, like the one in your arm. So basically they're going to be like this, and then a little clammy looking thing, and their body is in here, and they have these feeding structures that are like sort of tentacly. They're not tentacles with stinging, but they can capture food. So this would be on some sort of substrate, and this would be like the shell looking part. Barnacles look a little bit like that, but they're crustaceans. But these are, I call them a clam on a stick. They are different than clams, but you can see here the little clammy part and that sticky outy part in the middle. That's the lophophore is what we call that. So that's part of where lophotrochozoan word comes from. Some of these groups have the lophophore. It captures food basically and brings it into the body. Stick or maybe stalk, I don't know. Looks like a clam on a stalk. While we're here, because we don't need to know too much. Oh, and I guess they have a U-shaped digestive system, which makes sense with their body, that you'd have to have it not go through because they have a shell, so it comes in and out. You just hear people talk about that as a defining characteristic of synapomorphy, or at least, yeah, similar to both these groups, actually. Well, that's kind of what I got for you for Brachiopoda. Then we have this group, which we call, the most recent name is the Ectoprocta. But a classic name that people would talk about was the Bryozoa. So you might hear people still refer to that. It's an older name. These guys are colonial, and you can see again that they have these lophophores. These are really good pictures. So there's a number of them living together. They can live in different sorts of structures, but this is clearly those lophophore feeding structure there. Again, the U-shaped gut. Yeah, and so they're quite generally quite small, and they sometimes look like little snowflakes, or moss even, because when it's dried out, it looks like moss, but that's just the outside, just like the exoskeleton type part. These guys have, the inside is where the actual animal lives, but they call them moss animals because when you just see the dried outside parts of the colony, it can look like, kind of like moss, which of course is a plant, but oh, and these guys, I should mention, some moss animals here, and the nickname for brachiopods are called lamp shells. It kind of looks like a shell on a lamppost, I suppose. I think that's where it came from. So lamp shells are the brachiopods. Ectoprocta are the moss animals. We've already marked, we'll mark what group they're in. So both of these are lophotrochozoan. Defining characteristics. So brachiopods, stalk with an attached shell, right? And their nickname are the lamp shells, or brachiopods instead of Brachiopoda, the actual phylum. You don't need to know any examples of those specifically. Ectoprocta are called the moss animals, or ectoprocs. Bryozoans, you might also hear. And if we look at them, we see that they have the only thing, not worms, not any of those. Well, they do have a lophophore feeding structure. That's also true of the Brachiopoda and the U-shaped gut, as I mentioned. Both those groups have those. And these guys are colonial. Colonial's not unique. I guess that's what these, the green ones mean. They're not unique features, but they are kind of defining features of the group, just not actual synapomorphies per se. But if I gave you the combination of them, you'd be able to match it with what organism it is. Whereas the yellow ones are, pretty unique to that group. Now we have Annelida. So we've seen a lot of this here. They are in the Lophotrochozoa as well. We've seen this because we've done our dissection of the earthworms. So we can refer to these as, if you remember, we had those septa between each segment of the earthworm. So these guys are segmented worms. Annelid, or that first part of it means little rings. And that's talking about the segmentation because it has little rings around its body. And the other name would be, annelids would be like a common name of Annelida phylum. So we can look up here. I'll just gather these together, or you can look at your side. So what are we looking at with this? What did it look like for our earthworm? That's an example of an Annelida phylum animal. What do you think? Look at the chat. Need some help? Well, for sure with our earthworm, right? We had a worm body plan for these guys. Oh, I got somebody. Worm body plan segmented. That sounds like a good idea. Yeah. Because it had all those little, it's called segmented worm. Plus it had all the little, sections with those septa between them. Some examples here. We're just doing examples. I don't need you to know like the official names for these. Earthworms are an example, leeches, and these things we call polychaetes. It's a big group where they're not all like genetically related to each other, phylogeny-wise. They might be more closely related to earthworms and stuff, but you would hear polychaetes, and these guys are often in a hard tube, not necessarily, but often in a hard tube. They can live really deep underwater. They stick out of it though, because they are a worm inside of that tube. So you might hear something like a fan worm. In the book they show, or in the packet they show, a Christmas worm. It looks like a Christmas tree when it comes out. So it can come out of its little tube and then it fans out into a Christmas tree, why it's called, or looking structure, why it's called a Christmas worm. We also have spaghetti worms. Which the end of them, so they have a body, but then the end of them, and this is true of the Christmas worm, they have like a puff, like a Christmas tree looking thing, versus the spaghetti worm has all these little spaghetti looking things at the end. And all those are for, going to be gills for gas exchange, primarily. Might also help them capture food, but that's what they're going to, I think would also potentially help them capture food, but that's what they're out there for. And if they're polychaetes that move, they, which not all of them are in tubes, some of them swim around. Some of them are like jelly-y almost, especially if they live deep in the water. Other ones are like hard, not hard, but, full bodies, if you will. They'll have these little sticky outy parts on the side, which are for gas exchange because the gills are there, as well as for them to move around, unlike the earthworms, which just, crawl around, and leeches, which can sort of swim and also sort of crawl, but mostly swim, as they live in the water. Leeches and earthworms. So earthworms, right? So you saw in your ELN their benefit for composting and that kind of thing, and for making the soil both aerated and fertile. Also used for fishing bait. Leeches are they study the anticoagulants in them to that don't allow clotting of blood, right? So they might do that. Hirudin is what its name is, but anticoagulants. Also can use leeches for fishing. What else? I mean, they can use them to try to attack, I don't know, they have other ways of doing it, but historically, and sometimes maybe they use them to help attach organs back on. Like if you cut part of your finger off, maybe they would use a leech to open up, because it would secrete that anticoagulant, which maybe would help open it up before they sew it on. I don't know if they do that commonly anymore, but definitely used to be a thing. It used to be a thing to put leeches on your body if you were, sick, to get rid of your bad blood. Stick leeches everywhere to suck the bad blood out. So that's interesting. Could be detrimental, but that was a strategy historically. Okay. Then we go to mollusca. You can see it spelled with a K. You might see that in some books, but I'm going to spell it with a C because that's what our book does. Mollusks is the common name. They are also in Lophotrochozoa, Spiralia. Mollusks are quite diverse, so they range everything from octopuses and cuttlefish and squids, nautilus, all the way to snails and slugs, both living in the water and living on land. And then they also have the bivalves, the ones with the, have a two-part shell, like clams, oysters, scallops. Let's see, what other ones? Mussels, all those kind of things. Obviously, those are good foods for a lot of, that could be a good food. Some people eat snails, but also the cephalopods, which are the octopus and the squid food. Gastropods food. Abalone, people eat that. That's a gastropod with a shell, sort of like a snail, but the shell is kind of flat on the bottom. It almost looks like a half shell of a clam or something, but has a little bit different structure, and it's only one. So abalone is in there too. So those are good economic things for the mollusks. So what defines a mollusk if they're so diverse? And then there's also these 8-plated polyplacophores that we'll write in a minute. They have a visceral mass, which is like a chunk of tissue that contains most of their organs in it. They have a few different features here. So the visceral mass is a mollusk, special mollusk feature. So that's a synapomorphy of mollusk. Organs are mostly inside there. A few of them might be outside of that mass. They also have a foot. So it doesn't look like our foot, but it's how they move. So the foot can be like their actual body they crawl on, like a snail. The foot can be split into arms, like an octopus or a squid. The foot can be real hard in the side of a clam. So in the clam, when you take out a clam or an oyster and you eat that, you're eating the visceral mass with most of the organs in it. And then a foot, so if you eat those ever and you pay attention to them, you'll notice there's like a hard part at the end. That's the foot, which it's like attached to the visceral mass, but it can sort of stick out and help it bury in the sand to protect it from predators, or for it to stick out and then be able to pull in its food through filter feeding. And then the last thing these guys have is the mantle covering the visceral mass, which it comes in a lot of different kind of forms. But the mantle is basically like a draping tissue. So you got the visceral mass with the organs, and then you have some sort of sheet. It's like a sheet of tissue that covers it. So whether it's on the inside of the shell, like a bivalve would be like a sheet covering it. It flops over it in a squid. It's going to be the part that looks like the head, but it's on the top, but it's actually the head's down here, and that top is the visceral mass covering the organs. Same with an octopus. The part that you think of as its head is actually its visceral mass. Its organs are up in there. And then the little part with the eyes, that's really the body, and the head part with the eyes in it. Visceral mass of a sea slugs and sea snails, or water, ones that live in water, would be kind of like over the visceral mass. On land, they've been modified sometimes into lungs type structures, or like a pouchy thing that it can use to do gas exchange on land. So modifications there, but all of them have those three features. So let's look at the groups. You would want to know these official names for the groups. So we've got the cephalopoda or cephalopods. So these are not a phylum. This is like a class, kingdom, phylum, class. These are classes. It's actually more classes of mollusks. We're looking at 4 of them. So cephalopods are the octopuses, and it is octopuses technically because it's Greek word, which is not a Latin word. Latin word would be octopi, but it's actually octopuses. People say octopi now, and they've sort of accepted it, but octopuses is correct. Squid, which is both singular and plural. You don't really say squids. Maybe you can, but I don't know. Usually you just say, well, I guess you can say squids. Okay. Squids, the nautilus, which is the only one of these that has a full shell. So it sort of looks like a squid inside a shell. It's this beautiful chambered shell I can show you in class. And then at the end, it has its arms sticking out of it and it swims like that. So nautilus. And we have the last group, which is the cuttlefish, which I really love cuttlefish. There's a really great video. So I don't know if you've true facts, so there's this video, like a YouTube channel that's like true facts about the cuttlefish, true facts about the nautilus. And it's very tongue in cheek, a lot of it. But it's true. It's all true. They just do a very funny job. So Z Frank, Z-E, I think, Frank, or the letter Z. Anyway, he's the guy that does it. I'll put some in the board because they are quite entertaining, but also informative. So cuttlefish and octopuses, squid to some degree, but they're all very well adapted to being predators, all of them. They're relatively fast. These guys have closed circulatory systems. I'll put that in the other table, but we'll write it here because it's quite unusual. Most of these groups are having open circulatory systems. The other mollusks do, but it makes sense to have a closed circulatory system with cephalopods that really facilitates their predatory lifestyle because it can be more efficient at getting oxygen and gas around and food around, which allows them to move faster. They also have really well developed sensory structures like their eyes. They even have detecting eye things on at least octopuses on their arms. So they're not tentacles, they're arms. They also have all this, a lot of them can do color change of their bodies, so texture change of their bodies to blend in, which allows them to both not get eaten, but also to surprise their prey. And it's pretty cool. They do flashing things to ward off predation and maybe attract mates. It's pretty wild. So they are my favorite. I don't know if it's the cuttlefish or the octopus, but they're my favorite invertebrate. My favorite vertebrates are birds, but man, I like these octopuses and cuttlefish are amazing. Octopuses are also super smart. They can solve mazes and puzzles, and they're just really interesting if you look up stuff about them. Another group are the bivalves. So bi meaning two, valve meaning shell part. Valve is actually swinging doors, so bivalves, 2 shell parts. So there's a lot of groups in here. You probably know a lot of them. We got clams, we've got oysters, we've got mussels. It's spelled like that. We have scallops. Those are some good ones that are common. We've got the gastropods. Gastro means stomach, pod means foot. So these are the stomach foots. Oh, I should mention, cephalopods mean head foot. So the foot is attached right to the head. Gastropods means stomach foot, which is the foot is underneath the, it's on the ventral side, it looks like it's on the belly. So that's where the gastropod comes from. So these are going to be snails and slugs, both living in water and land. Slugs have lost their shell. Snails still have them. They can live in both. They are adapted to their environment. Obviously, the ones in water are going to have gills. The ones on land have other kind of gas exchange structures, like I said, the modified mantle for a lung sort of thing. They do have to stay wet, though. As you know, they'll dry out pretty quickly. And that's because a lot of gas exchange, or some things are across the body wall, and they'll lose the water there. And then the last group are the polyplacophores. Might also hear them called chitons. I don't know, they don't really have another name. But they have like 8 shells. So it sort of looks, it's like a shell like this. Their sluggish body is underneath. And then they have a shell like this and it has like 8 plates on it. So they crawl along. Polyplacophores. I think they live in water. Don't quote me on that. But yeah, so they have a shell on top with just 8 plates. And there are, I think there's, I want to say 7 or 8 groups of mollusks, but these are 4 that we are worried about. And you can see a lot of these are foods. I know that they study some snails because they have toxins in them. Actually, if you look at a bivalve, sometimes you see the dead bivalves, meaning like just the shells, and you might see a hole in it. That hole is likely caused by a gastropod that can drill into the bivalve shell and then basically put a toxin in there or some other mechanism that actually like can paralyze it or whatever, and then it can get into maybe it even causes it to open, but it can eat it from there. So that's what those little holes are typically that you might see in a seashell, particularly a bivalve seashell, like just the one valve of it or something. Okay. Oh, chitin. Sorry, not with an I, with an O. Chitins. Different than the material that makes exoskeletons. Now we get to the ecdysozoa. That's why these are separated. So we have Nematoda, Tardigrada, and Arthropoda. Arthropoda is the most diverse group of animals by far. But we have the Nematoda or nematodes. They are also called the roundworms or nematodes. Now they are an ecdysozoa, so we'll give some color there. Okay. Now, ecdysozoa do ecdysis. So ecdysis is like shedding of the exoskeleton or outside cuticle. So all these groups, when they grow, they have to molt. Basically, they get rid of their outer layer so they can grow bigger. That's what ecdysis is. That's their kind of like the group's defining feature. It's also molecular DNA stuff. Uh-oh, board. Okay. So ecdysis, the exoskeleton or cuticle shedding to allow growth, basically. And crustaceans are an arthropod, so you know perhaps ecdysis, like that idea of soft-shell crabs. They've just shed their exoskeleton, like the outer layer. It's not like they lose their whole shell. They do the outer layer, and that's why they're soft, because it hasn't been calcified yet. You probably have seen cicadas in our area that like you just see the exoskeleton, you're like, and it's split open the back on a leaf and it's stuck there, and you're like, where is that? Basically, it's come out of, with its new layer, it's come out of and shed its old layer, did ecdysis. If you've ever watched caterpillars, they shed their outer layer when they grow. This also happens with the nematode worms. So nematodes are the roundworms. So if we go up here to our descriptors, pull these there. We have only one worm body plan left. So that's the worm body plan with the tapered ends, no segmentation, not flat. So that's your defining characteristic, basically. And genetically, they go together. They can be very small and microscopic, like C. elegans. I can write that in an example. That's a very important, we call it a model system. So C. elegans, like E. coli, it has a full name, Caenorhabditis elegans, but everyone calls them C. elegans. That's its actual species name. And the reason they're important is obviously all of these are being researched for their own selves, but they use C. elegans for research, particularly for cell fate and developmental biology stuff, nervous system kind of things. So C. elegans are cool because you can kind of like tag every cell, and they have programs where you can track every cell throughout all of development until they become a worm. So it's like, you know, the parent cell in the early embryo and where it goes and exactly what it turns into and who dies. And it's very consistent about exactly what cells do what. And then you can manipulate the system and see, well, what happens if this cell does something different, or if this cell is taken out of the embryo, like what goes on here. So we do use C. elegans for research into developmental biology and I don't know, want to say that? Let's leave it at Dev Bio, because that includes nervous system development and all kinds of stuff. I worked in a C. elegans lab for a while. So it's a free-living flatworm. It's a microscopic one. Not flatworm, excuse me. Free-living nematode. Meaning it's not parasitic, right? And there's lots of other groups of free-living nematodes as well. This is just an example. Then there are the parasitic nematodes. So this is like going to be a number of things that live in soil and infect your plants. So often there's a lot of reasons your lawn might go brown or yellow, but it can be a nematode parasite in the soil that's affecting the plants and basically infecting them and killing them. So you do see this sometimes. There's like classic patterns of the grass. I feel like it's in a circle. I'd have to look, but I feel like if it's in a circle of yellow or brown, your grass is dying, it might be a nematode infection. There are some nematodes, the free-living ones in soil, though, that are essential for plants to live. So not all of them in soil are bad, but there are some parasitic ones. So let's see, how do we want to say this? Some that are in soil that infect plants. We also have ones that infect animals like humans. So you might have heard of pinworm infections. That's an intestinal infection. And like I had a friend who grew up, well, I mean, people get it in the US too, but it can be more common in other places. And she grew up in India, and she was telling me that when they were little, the parents would go around and check for pinworms at night, because I guess they come out kind of at night. And so they'd have to go look at the little kids, you know, like their like anus, because that's when they could see the little pinworms and they'd know if they had an infection. And then you can get treated for them. They have treatments, but they are an intestinal infection. We also have hookworm. Hookworms get in through the feet, most likely. So, you know, sometimes your parents or, you know, whomever would be like, wear shoes, don't run around barefoot. And you could get hookworms through the foot. So that would be an infection there. We also have heartworms. So, you know, that's a big industry for dogs, right? In particular, I don't know. Do cats get heartworms? Does anyone know? I know for sure they give heartworm medication and shots and stuff, or preventative stuff for dogs. Maybe cats get them too. I don't know. They're worried about them in pets, obviously, because you don't want your pet to die. But that's an infection, like the worms get in the heart, obviously, and they can infect the heart there bad, right? And those are transmitted by insects, I believe. So you can get heartworms. So that's a big industry, right? Prevention and treatment, perhaps, of heartworms, and then these other parasites. So you could use those. And I think the heartworm would count as different than infections in humans, right? Because it's very much different with the pets and the vets and all that kind of thing. Then we come to tardigrada or tardigrades. They have a couple nicknames. I don't want that. So you could call them, oh, someone tell me about cats. Google says cats can get them, but more resilient than dogs. Interesting. I think I'm correct, though, that mostly it's all about the dogs getting the heartworm prevention medicine. I feel like that's what the commercials look like, at least. I don't have a dog, but that's what I think. So tardigrada, or that's the phylum, tardigrades. Thank you for that information. So tardigrades, you can also call them water bears. Sometimes they call them moss piglets. They're microscopic, pretty much kind of brownish see-through. So these are ecdysozoans, so are the arthropoda. Obviously, I grouped them together in this little set. So these tardigrades are really interesting, and they study them because some nematodes can do this too, like particularly C. elegans. Let's put that in there. So it's not just developmental biology. They also study aging, because these can go into a suspension state. It's called Dower, D-A-U. ER. So they go into the suspension state, and they can like stay in this state. It's almost like an endospore idea in bacteria. So they can stay in this state in unfavorable conditions and basically survive for a really long time, and then come back, and then they like just are okay. So C. elegans do that, so they can study aging, and then also, I mean, other things related to aging, but like how do they do the suspension state? Same with the tardigrades. So this is not a subgroup of tardigrades, but maybe I have this in the characteristic. I do. Some can survive in very harsh conditions in a dormant state. That's actually also true to some roundworms, but that's classic tardigrade thing that they study. They're the ones that they like send into vacuums. Like, let's send these to outer space and see what happens. They put them in vacuums and they're fine. They're like wildly resilient. So very interesting to study in this dormant kind of state, which their body completely changes and then comes back. And these are even more harsh conditions than C. elegans. But like C. elegans, if you take away their food and starve them, they'll basically go into dour, and then you put food and then they come back. So tardigrades, that's why people are interesting. Unless they're really cute, you can actually just see them in moss. I don't know if we'll have time. Maybe we can collect some moss and see if we can see some tardigrades. So they're not super rare. They can live in normal conditions. They're not just in crazy places. But this kind of survival thing is very interesting for people to study. And they are pretty adorable. Microscopic. This is a scanning electron micrograph. There's other pictures that are even cuter. And they have these little clawed feet. That's a classic characteristic too. Got those little clawed feet on there, these soft bodies. But they do shed their cuticle, I believe, in ecdysis. Claws and cuticle, that's going to go with them. And this is, let's change this to green because it's not unique, but that is a feature. And really the harsh conditions is not entirely unique, but it is a feature. Oh, I forgot segmented. So segmented, we said segmented worms were those annelids. So let's go back to Annelida and put segmented in there too. I should mention segmentation can be an advantage, or is an advantage, maybe I should say, because you can have specialized regions. So similar to like having a complete gut, segmentation can kind of afford you to that in your whole body. And then I'll go to insects. Yeah, perfect. After this. So segmentation, annelids have segmentation. The arthropods we'll see have segmentation. Segmentation also used to be used as a feature. So they grouped annelids with arthropods because they're like, oh, they're segmented. That's a synapomorphy, but it turns out it's not. Genetically, it's clear that they're not. Segmentation is the last column there. And this actually gives you a lot of interesting information about who has ventral nerve cords. Dorsal we'll see is only in deuterostomia. Through gut or anus, and that means that it has the complete gut, like the complete gut, right? The alimentary canal. Mesoderm is going to be all those bilateriates, right? Because that's the third tissue layer. Okay, so but segmentation though, Annelida. So that's one that has segmentation. Then you have this group that we're not talking about. Kineranka. And then you've got these groups here, Arthropoda, Onchiophora, which are the velvet worms they do talk about in the books, and Tardigrada, they're talking about segmentation too. So you can see segmentation is spread throughout the tree, so it's basically not a feature you'd use to group them, but it is a descriptive feature. And I believe this light blue means some are segmented, but not all of them necessarily, even though annelids are called the segmented worms. So what's the advantage of segmentation? And I guess we can add segmentation to Tardigrada, now that I see that. Specialization along the body. So you can have specialized, like you saw it in the crayfish, right? You had specialized legs on each, in the different sort of regions or segments. You didn't see it quite as much in the earthworm, but you did, see that each segment had its own unit of organs, really. So specialized regions of the body because they have different structure and therefore different functions. They also have more flexible movement. And if you think about an earthworm moving or a leech wiggling, clearly a lot of flexibility there. Same with the arthropods. And they, like think of the crayfish tail or shrimp tail. It has segmentation. It can even bend, even with its exoskeleton, which is really kind of important because the exoskeleton is quite hard. So if it was not segmented, you'd really have no flexibility at all. It would sort of be stiff like a coral is, the coral or the tube worm perhaps. So although that is an annelid, but I don't think it has segment. Not sure about that, but for sure in the crustacean type group or any of these groups that have the segmented exoskeletons, it allows more flexible movement, which could make you be able to move more quickly or more specialized movement. So with the segmentation too, it's like in the insects at least you have like segments that have the wings, and this whole segmentation business too is the Hox gene stuff. So Hox genes set up the segments and they set up the specialness of each segment. So which Hox genes are there dictate what that segment is going to be and how it's going to look and what it's going to do. So we have the Hox genes involved in this segmentation here. And this is also the complete gut is relatively similar, but not the flexible movement. That's just the segmentation. So we do have that. And then we come here, jointed appendages is the synapomorphy of arthropods. And we know that arthro means joint, like arthritis. And we know pod or poda is foot or leg, could be leg. So these have jointed legs, which means, they have bendy parts. And you clearly know this from looking at your crayfish. Who had the gastrovascular cavity that I missed? Platyhelminthes. Sorry about that. There's basically 4 kind of main groups. Let's see a time. Okay. So we're going to do it like this. We're going to do it in quotes because crustaceans are not a true group. Maybe I'll draw a little phylogeny to show you, but we have like crustaceans. So classic crustaceans, they're not actually all grouped together. Some are more closely related to insects than they are to other crustaceans. So it's like crustaceans nests with insects. So if you wanted to count multiple groups as crustaceans, you have to count insects as crustaceans, which is weird, of course. So they kind of call it pancrustacea, which means insects and classic crustaceans. But crustaceans traditionally are things like our crayfish or crawfish, lobsters, crabs, shrimp, little krill, barnacles, what else? Roly-polies, which are isopods. Oh, I'll look it up. Or you might call them pill bugs, that kind of thing. They're maybe the only crustaceans. They also call wood lice. I think they're the only ones that live on land like all the time. We also have sea monkeys. I don't know if you can buy sea monkeys to you pour this powder into water and then they turn into little sea monkeys, which are these little crustaceans and move around, very small though, but you can see them. It's like a kid's thing. They do. A lot of times you dye the water so you can see them because they're kind of clear. That's what I have. There are other groups as well, like some that live in most of these live in the water. Those are classic ones though. Insects. Big important group here, most diverse group of everything. They're going to be things with, often they have wings. If they don't have wings, they probably lost their wings. So often they're going to have wings. 3 body segments. I guess we'll write this in a minute with the insects, but they've got 3 body segments. They've got 6 legs. 6 pairs of legs. Not 6. 3 pairs of legs. 6 legs total. Yeah, hexapods. So anything you're seeing with that. So, what do we have? Bees, butterflies and moths. We've got flies, We've got ants, we've got beetles, the most diverse group. All kinds of things like that. Fleas. Roaches, which are like along the beetles lines. Then we have what we call myriapods, meaning many feet. Myriapods, many legs. So these are going to be centipedes and millipedes. They do have many legs. Centipedes have 2 pairs of legs, or 1 pair of legs per segment. And millipedes have 2 pairs of legs, which just means each segment has 1 on each side for centipedes, 2 on each side for millipedes. Millipedes, so centipedes are, they'll bite you. They bite. They have like little fangy things and they can hurt or even give you like an infection or a poison kind of thing. Millipedes, on the other hand, don't. They eat detritus, they, dead leaves and stuff like that. So if you see one and you're like, ooh, is this segmented wormy thing with legs, which is a myriapod, a millipede or centipede, look at its body segments. If it has 2 per body segments, a millipede, it would be safe to move. If it has 1, don't touch it. So 2 pairs per legs per segment. Okay. They don't bite. And then the last group there we have, so I could ask you like the difference between millipedes and centipedes and to know that they're myriapods. Could ask that. Chelicerates, so it's a K sound. So chelicerates include the horseshoe crabs, and they include the arachnids. Arachnids are spiders, as you probably know, but they also include scorpions, also includes mites and ticks. Chelicerates have these chelicerae. They're like pinchy things at the front. It's like right around their mouth. They've got 2 chelicerae, a pair of chelicerae on them. I know people think spiders are insects, but they're not. They're arthropods, but they're different than insects. Most arachnids, some might be slightly modified. Most have 4 pairs of legs, which is your 8 total legs. Unlike the insects, which have 6 total legs, 3 pairs, these mostly have 4 pairs of legs. So 8 legs total. And let's, in the last bit, let's do, and there's nice pictures of arthropods here. So some diversity. Here are the chelicerates. So you can see clearly these little, see these like dark ones right here, or here on the scorpion. So this is the front, those are the chelicerae. And you can see them on the spider, these middle ones actually. These owl ones I think are the pedipalps. Maybe vice versa, but 2 of these, I'll look and label it. 2 of those are the chelicerae and 2 are pedipalps, which are a little different on spiders. But and then those insects have those 3 pairs of legs, and these guys have wings underneath their shells, 30 pairs of legs versus the 4 pairs like you'd see on arachnids. You can see that's true here, 1, 2, 3, 4 for this tick or mite as well. Mites and ticks obviously can be vectors of diseases, particularly ticks. We talked about them transmitting Borrelia burgdorferi it's a bacteria that gives Lyme disease, so that would be something to think about economically. Spiders make the silk, which we can use to study for tensile strength and making our own materials because it's very, very strong. Mites can also, like dust mites, right? So those are little animals, like little animal mites living in the dust. They can activate allergies in people. Allergies are often also activated by little like cells and stuff. So it's not always mites, but dust mites can be something that bothers people. And then of course you have to watch out potentially of getting bites from scorpions and spiders. Could be poisonous or hurt. And like I said, I'll fill in this little table for you so you have it all nice and together with the A&P stuff that you'd want to be kind of familiar with. It's like mostly comparisons between the groups. Okay, so with the insects, they're going to be primarily on land. There's a few groups that live in water, but most insects are living on land here. So there's an essay about this. Challenges of living on land. We mentioned one got to worry about your body drying out because the gas, with the air is going to make your water evaporate. So how do you deal with that? Or what's an adaptation that'll allow them to not dry out? And I would usually ask you to come up with some ideas, but in the interest of time, I'll just come up with them. How about supporting your body in gravity without the water buoyancy? BUO, sorry, that was wrong. So if you're in water, you've got that buoyancy, which counteracts gravity, right? But on land, you don't. You just have the gravity pulling you down pretty much. So how do you deal with that? What's an adaptation there? You have to worry about the eggs and sperm drying out. So not just the body. You'll notice a lot of these are the same things plants had to work with, right? Or the plants had to have adaptations that arose to allow them to move to land as well from being green algae in the water. If you're not in water, you're not going to just have sperm just swimming around or your eggs just being dispersed like in water. You can just release the stuff into the water and sometimes it's deposited into or near the female the sperm might be, or in the same place. They might deposit in the same place, like sperm on top of the eggs. But once you get to land, you really got to be like, well, how are we going to get these dispersed? Because they can't just float through water or swim. What about movement? The other groups are swimming with maybe those parapodia that stick out on like segmented worms like the polychaetes, or could be by the undulating body in the water, or like a flatworm or a sea snail or sea slug. Could be the arms with that kind of motion by the squid and such. But now on land, it's going to be mostly things like walking or maybe crawling, but for insects, it's going to be like walking, it's going to be flying, it's going to be jumping, those kinds of things. You've got to do some kind of gas exchange, especially gas exchange without drying out your body. So specifically in air, right? An advantage of air, though, on land is that actually in land you have, or on land, in air, I should say, you have a higher percentage of CO2 in air than you would in water. You do have oxygen in water, obviously, because the organisms need that for cellular respiration, but it's just when it's dissolved, it's at a lower percentage. In air than in water. Disadvantage. Gas exchange needs a wet surface, the diffusion, or moist at least. So how do you keep that moist? General strategies: keep it inside the body. Live in somewhere wet or moist, or keep the gas exchange surfaces inside the body. But then you have to have some way to get the gas in, like openings in the body wall, tubules, that tubes, that kind of thing. And then we've mentioned this a little bit, but they've got to excrete nitrogenous waste without losing too much water. So that ammonia is not going to work at this point, or on land, for the most part at least. And in a less toxic form, right? Because you don't have a lot of water to dilute that toxic form, the ammonia, in other words. So we can say less toxic form, aka not ammonia. I'll number these so we can. Okay. So not letting the body dry out. So what are some strategies that evolved in insects in particular in order, that allow them to not have these things be a problem? So, of course, it doesn't evolve for them to be able to move to land. It evolves and then they're able to move to land, right? It's not a directed evolution, but an exoskeleton evolves. So that's a big thing. Not that other groups don't have exoskeletons, the other arthropods do, but the exoskeleton does protect its body from drying out. That's mainly what insects use. So all the wet tissues inside this exoskeleton is on the outside. This is also kind of targeting number 2. We've got the exoskeleton with muscles attached to it. So the muscles attach on the inside. This gives a lot more power. To the movement, because with our muscles are attached, right, to tendons and ligaments, depending on what they're attached to on the other side. But this allows the pulling of the muscle, which allows a lot more power to be generated. So it allows you to move through on land without the buoyancy of water, and also do things like flying, do things like jumping. So that's some special insect stuff. So because of this muscle attachment, they can jump. Insects can fly if they have wings. If they've lost their wings, they can't. They can walk, of course. So they have lots of different types of movement. Flying is a big deal because you can get further away. Escaping predators, getting food, your little restricted area of where you can do your, general life increases. Might not be super far, but it's further than if you can just walk. Maybe, swimming gets you far too, but these kind of movements are pretty advantageous. Eggs and sperm from drying out. And fur, and this is 4 as well, basically. Lots of insects have internal fertilization. We're going to actually have some way to deposit the sperm into the female reproductive tract. And then the eggs would be right there, and then the sperm can swim, and then they can fertilize right there. In fact, insects have lots of very specialized penile structures or penises. They're like crazy shapes. Was an art exhibit where they did shapes. I can put a picture in here. But the reason, or the advantage, is that they're very specific for a female reproductive system of the same species. And as we know, because of barriers, if you have non-same species mating, it's just a waste of time because the egg and sperm are not going to be able to fertilize, most likely. They're very specifically adapted to only fit together. Like a corkscrew type penis is only going to fit into a reproductive system of a female that has thread-like things inside. It's quite interesting. So they're highly specialized that way. So internal fertilization is going to be an advantage there. And they also have shells on their eggs. Not that other groups don't, but they don't have jelly eggs a lot of times. At least maybe if they lay them. So that's also kind of goes along with this, but how do you keep the embryo from drying out? The fertilization would happen inside. Often you're going to have an egg casing or shell, though, when it's laid. Fertilization can't happen through the shell as far as I know, but it can be made. And that's a strategy. Some carry their young in sacks and things, but we'll get to that. Oh, did I do these out of water? Oh, supporting body, exoskeleton. I guess that's also and the movement one, number 5. Okay. So this is number 6, gas exchange. All right. So in insects, and this is also true of spiders, they live on land, which are not insects, but they are arthropods. But in insects, they're basically have their gas exchange structures on the inside. So they have what are called the tracheal system. It's quite different than how we think of gas exchange with lungs. Here, so here's the opening in the body wall, the spiracle. And if you actually look at an insect, you'll see these little tiny dots down the side. Those are the openings to the body. So we've got this opening here, the spiracle. Then you've got all these tubes, and the tubes branch, 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 branch. You can see that in this picture here, where you see all the branching inside the body. And then each cell has a little connection where the gas exchange actually happens. And it's not just O2 out, it's it's CO2 out as well. There's your spiracle. And then the gas comes out. And then there's little air sacs too to store the excess gas so the exchange doesn't have to go all the way all the time. It can store it, then exchange while the other one goes out and such like that. But everything's happening on the inside of the body. The nitrogenous waste, we already talked about that they use the uric acid. And I'll just say see other notes. In addition to the uric acid, it's one, and then we also have talk about Malpighian tubules. Malpighian tubules are a bunch of tubes that go into the body cavity. So it's on the intestine. So they're little projections off the intestine. You have water coming in here, water and ions really. So, and that's from the body. So it's in the coelom. It's a bunch of liquid because these guys have open circulatory systems. So all their liquids in their body. It does have a heart type structure and it drains back into that. We saw this in the crayfish as well, the open circulatory. So it drains back in and then just bathes, it comes back out and bathes the fluid and then goes back into the heart, get pumped around. But these guys don't have gas exchange. They don't have gas exchange with the circulatory system. That's separate. They have it with the tracheal system. But here we have water and ions from the body going in. They're going to be reabsorb a lot of the water. Then they'll get the feces out, or that are undigested food, and also the uric acid will come out, this opening at the end, the anus. But they're going to be retaining a lot of the water because they pull it back in, regulating the ions. So this is an osmoregulatory system in addition to the excretory system. So it's both nitrogenous waste as well as osmoregulation. So that's a big thing there, so they can retain that water. Osmoregulation and excretory system. So those are adaptations for that in insects. If we're thinking about insects as the most successful group of animals, as we mentioned, it's not just about adapting to living on land. We can have other things as well. So flight, we mentioned, with the ability to move in different ways. But then also this complex life cycle, so often metamorphosis in insects. They can have complete metamorphosis where the body forms are totally different in the larva and in the adult. So this is an example here. You've got this larva grub. A grub is a larva that's going to become a beetle type. And so you can see the grub looks extremely different than the adult beetle here. So we have like maggots, which are larval forms of flies. We have caterpillars, which are larval forms of moths and butterflies. The reason this makes them quite successful, this complete metamorphosis, is because they have really specialized functions in these two body forms. So in the larval body form, pretty much what they are specialized, and their body parts allow them to feed and grow. So they can eat and eat and eat really quite efficiently, quite quickly. That's what they do. Then their bodies undergo a complete change. So it's not just that they build something around themselves or their body turns into a chrysalis, which is a type of pupa kind of thing. Their body tissue of the larval stage completely breaks down, and they have these stem cell populations inside their body that totally grow into the new organism. So like the adult beetle, it's from totally new tissue. It's not just like a, I think people think that the caterpillar just wraps itself in a cocoon and then sprouts wings and turns into a butterfly, but no, that's not it. It's like the caterpillar actually usually turns into the chrysalis, its own body. Some might do a cocoon, but then the whole thing breaks down in a totally new organism, comes from the stem cell populations inside of the larva. And then the adult form is specialized for, in large part, some feeding. Some insects don't even feed at all as adults, but mostly breeding. So we call them feeders in larval and breeders as adults. And so this specialization for breeding or reproduction allows the insects to be very successful there. So it's the body doesn't have to do all the functions well because there's two forms of it. Now, some insects don't do metamorphosis. Some do incomplete metamorphosis where the larval stage looks very similar to the adult stage, just usually smaller. So, this is somewhat variable, but it's definitely a thing that makes a lot of insects very successful. Short generations as well, so it doesn't take too long to reproduce and make the offspring, and lots of offspring produced for each round of replication. Okay, I guess I have one more little thing I want to mention, which we've already kind of talked about with excretory system, particularly with osmoregulation, is this idea of homeostasis. You'd want to be able to talk about some homeostatic mechanisms. There's a really good, one of the masterings that you did talked about homeostasis, so take a look at that again to get some ideas there about homeostasis in these groups that we're talking about and how they regulate that. So first of all, you kind of okay, environment changes. It's never the same all the time. So what do animals do? And this is true for all organisms, but in particular, we're looking at animals, obviously. So what do animals do? Well, they can either regulate, which means they're going to control, they're going to keep the body in some way for some feature. They're going to keep it different from the environment. So they're going to regulate. So even if the environment changes, the body is for that feature, whether it's temperature, water content, ion content, the potentially the concentration of different small molecules like sugars and things. What else? Could be acidity or pH of the body. Lots of different things that it could be. But if the environment changes, it's going to regulate it to keep it pretty consistent, or at least different from the environment. So that's a regulator. Then you have conformers, which basically will fluctuate with the environment. And most animals will be regulators for some things and conformers for other things, right? So it's not it's not like a blanket, you're a conformer or regulator. It just depends on what you're looking at. So here's an example of a river otter, which is not an example that you'd use for anything in this unit and these are not examples because they're, vertebrates. They're not in Animals 1, they're Animals 2. But this is what the book has as an example of temperature regulation, right? So the river otter is obviously a regulator because it keeps, despite the ambient temperature, which means the outside temperature, despite that changing here, the body temperature of the river otter stays relatively constant. So it's a regulator. Versus temperature for this bass, this type of fish, you can see as the environment temperature goes up, also the body temperature goes up. So you can see that there's a relationship here where it's changing and then they're conforming to the environment. Now, is the bass going to be a conformer for everything? No, in this case, temperature, but it's not going to just let its water content go willy-nilly because then it could explode or dry out. So it's going to have, regulation for that. So when they're talking about regulators, so if you're a regulator for something, what it's doing is keeping the body in homeostatic state. Now, what I want to point out is homeostasis is not equilibrium. This is a very common misconception people have. Equilibrium means that equilibrium would be more like a conformer, honestly, where it's like the same externally and internally. That would be like equilibrium. Regulators and homeostasis are really like mostly non-equilibrium. All that it means, homeo means like same or steady. Stasis is your state. So you're doing a steady state, which basically means you're having a particular set point or like a range. So that's how I think of it. Homeostasis or homeostatic mechanisms is the tendency to keep an internal condition in a certain range. So a small temperature range that the body stays at, a small water concentration, like body concentration, a narrow pH range, a narrow glucose concentration range. All those things would be homeostatic states. So you're keeping the body in homeostasis. And this is one of our learning outcomes for the course that is important. So homeostasis is critical, right? So you have feedback mechanisms to keep it at homeostasis. Like when something gets too low, a body system and process kicks in to raise it up. When something gets too high, a body system kicks in to bring it back down. So they give a thermostat example here. Like if you had it on a set temperature, it heats up or cools down. But the body systems, of course, are going to like retain more water or like lose more water. It's going to retain more ions or lose more ions. It's going to do something to get the body temperature up or do something to get the body temperature down. A lot of the groups we talked about now don't regulate their body temperature. They're thermoconformers, where they just stay with the body temperature of the environment, which can restrict them to certain places, but maybe not. And everything has different set points. So it's not like room temperature is the best for everything. I mean, it depends on where they're adapted to live. pH 7 is not the set point for everything. Some stuff, but other things like our stomach pH is pH 2, right? Like that's the set point for the stomach pH versus the blood pH is a little higher than 7. So that's the little higher, a little lower. Around 7, a little bit different than 7. So that's the set point for the blood. So even within an organism, you can have different set points to keep homeostasis or homeostatic conditions in those regions even. So for example, osmoregulation, the regulation of water we've talked about. We've got osmoconformers, which are isoosmotic. Iso means the same. So basically, whatever concentration the environment is the concentration their body is. Lots of our groups are like this in this unit. And then osmoregulators are going to regulate how much water there is in the body, expend energy for that, and they'd be hypertonic or hypotonic, just depending on their needs. So hyperosmotic, I should say. These are similar to hypertonic, hypotonic. Hyper means higher or more, and hypo, as you recall from Bio 1, means less or lower or under. So hyperosmotic would mean that they have a higher concentration of something than the environment, and hypoosmotic would mean they have a lower concentration of the solute, which is also related to like how much water there is. So the lower concentration of the solute would sort of mean like it has more water pressure inside or less water pressure. So things like what are going to be regulating this is all those osmoregulatory systems. So the Malpighian tubules. In the insects, we've got the Protonephridia. In the flatworms, we have the Metanephridia. In the annelids, those are the big ones we talk about. We have just Nephridia. In stuff like mollusks, do similar kind of thing with the regulation of the water. Sometimes they even call them kidneys. Different than our kidneys, but most of them have tubules, and the water goes in, and then the water comes back out in different amounts, depending really on how many aquaporins they have on their cell membranes, and they can regulate that. And there are some other factors as well, but aquaporins are generally how you get water across a membrane in large amounts. So these are insects, Protonephridia, flatworms, or Platyhelminthes. Metanephridia, Annelida. Nephridia. And I'm writing the phylum, so Arthropoda insects, Platyhelminthes, Annelida, Mollusca. What else do we have? Oh, in our crayfish, we had the green or antennal gland. So that's an arthropoda. Crustacean, crayfish. So different groups have different things. But you particularly would see osmoregulation for stuff that lives on land. A lot of the ones living in water are osmoconformers. So these are going to be living on land, or at least partially living on land, like the crayfish can. The mollusca with the nephridia, like let's say land snails. I'm sure the other ones have regulatory systems, but it's just not going to be as regulated. There's temperature regulation. Like I said, there's pH regulation. A lot of what we talk about is the osmoregulation, though, because we have specific examples there. So that'll be that then. And, some people do eat insects, so you can come up with some of those examples. Insects are also pollinators. Do people actually buy and sell insects? Well, they do bees, right? People do raise bees. That's a money thing. That's economic. And they can use those bees. Sometimes people raise the bees because they actually need the pollination because they're trying to get honey. And bees would, they produce the honey. It's not from the actual plant. So maybe you'd have to put it in the context of how it's being used by humans, not just that it's a side effect. So just make sure you're armed with a number of examples that are highly diverse of that. Oh, bivalves, right? So bivalves are the things that produce pearls. So you can have mussels producing pearls, oysters producing pearls, clams produce pearls, a lot of scallops produce pearls. And actually what that is that a little grain of sand gets in there between the mantle and the shell, and it's basically irritating to the bivalve. And so it secretes this shell, the nacre, N-A-C-R-E, which is that inside shell stuff to cordon it off so that it doesn't bother the body anymore, and turns into this little ball because it puts a number of layers around it, and then that's what an actual pearl is. And obviously those are sold and bought and can be quite expensive. So that's an interesting thing. That's Mollusca phylum, particularly the bivalve group within the bivalve class. And then, some of those, you can give some examples of that. So that's something as well. Yeah, so be creative with it, but, make it actually not a super stretch, right? Something that people are actually buying and selling that's making the economy go round in some way.